we also call fixed differences. And later, there are model-based approach, among which T1 statistic is analogous to HK, which also examines the ratio of polymorphism to substitutions. And in addition to that, T2 also considers allele frequency, not to be confused with the T statistic introduced earlier. More recently, there are novel summer statistics being developed specifically for balancing selection. And in one way or another, they both aim to detect the enrichment in allele frequency. Now, all these methods face a common constraint, which I call window size conundrum. So let's go back to the same sequence, and for some reason the color's a little messed up, but just so you know, there's a region of high diversity around that side. If we started off scanning this sequence with a small window, close in size with what we expect the footprints to be, then yeah, we should have pretty nice power to pick that up. However, when we do have a small window, inevitably there will be more noise, and across the genome, the footprint size of balancing selection would be very different. So it's hard to justify using one same small window to scan across the whole genome. If only we can use a large window that has less noise and be downward compatible with more footprint sizes. But as you can see here, whenever we use a large window, everything averages out. And this is not only the case for summary statistic. The current model-based method also faces this constraint, and let me show you why. So for the T1 and T2 statistic developed by Giorgio back in 2014, it's a set of likelihood ratio statistics that's essentially asking how much more likely the data we observe fits what we expect under balancing selection versus neutrality. But the model they adopt for balancing selection is the one based on coalescent theory for its genealogical model. And instead of also adopting a coalescent-based neutrality model, the authors chose to use the genetic variation level for the whole genome so that they can better capture factors like demography. But this trade-off also means that the model behind their alternative hypothesis here do not assume the same neutrality as the actual neutrality used in the statistic. And as they integrate more and more data points in this statistic, more and more bias will be introduced and eventually overwhelm the true signal. And as I will show later, this also compromises their performance under oversized window. So to tackle this problem, we turn to mixture model, and here we also take the genome-wide variation level here, the site frequency spectrum, as our neutrality. And for any site under balancing selection, we model that the probability distribution for the influence of balancing selection be binomially centered on the equilibrium frequency of the selection. And then for any neutral site we have that are d distance away from the site under selection, it's the probability to observe its frequency would just follow a mixture of neutrality and the influence of balancing selection. And we make the mixing proportion alpha d here as inversely related to d, so that the larger the d, the smaller the alpha, just like how linkage would decay. And the resulting likelihood ratio we see here would be, for the full model, we maximize that over the parameter space. And for the null model, neutrality, it is also a special case of full model. It's just we are taking the alpha as 0. And with this approach, we made 5 variants of this statistic, each tailored for different type of input data, regardless of whether you have whether you have substitution costs or whether you have polarized allele frequency or minor allele frequency, you can always find a variant that can be used. And collectively, we call them B statistics. So to evaluate the performance, I simulated a bunch of sequence undergoing very old balancing selection and applied the method that I mentioned earlier with varying window size here shown on the x-axis. And the y-axis, we just beat their power. So first, if you look at the summary statistic, apparently when we use large window, their power decay very fast. And for the model-based T statistic, because they incorporate the locations, their performance improved, but still, if you look at even the best performing T2 here, over half of its power is still lost. And but if we for simplicity, I will only keep the model-based method here and only show the analogous method, analogous B statistic here. So if you look at B statistic, however, not only the highest power it has, around almost the same as the T statistic, it also has way minor decay and it's stabilized at a pretty satisfactory level, and we think that's great. And next, I went on to explore how it performed for different balancing selection of different age or equilibrium frequency. And for that, I applied B statistic with all the data on that sequence, because that's how we want to apply it when we apply it on empirical data. But it become a question in what window we choose for other statistics, because they actually don't perform well if we want to match the data they use. So instead of matching the data, here I give them the optimal window we can use. And here, this is when the equilibrium frequency is 0.5, and later you see for other equilibrium frequencies, the power of B statistic actually improves, and at some point even outperform T statistic when they're using the optimal window size. And we think that's great, and at least so far we know that B statistic are able to detect balancing selection powerfully, even though it's being applied with the least optimal window size. And next, I went on to apply them on empirical data, and for that, I choose bonobo because I think they're cool. So bonobos and chimpanzees, they are one of our closest sister species. And they both live in Congo area. It's just that bonobos live across the Congo River from chimpanzees, where there's lush forest and food is more supple. And unlike chimpanzees that are very male dominant, bonobos have matriarchal societies, and they rarely have violence to their own kind, and they're very fun-loving. They also have very high empathy level to their con-species. And one of the most idiosyncratic feature of bonobo is that they have these social sexual behavior, which they use for socializing. They use that for bonding, tension relief, favor exchange, etc. And because it's socializing, both males and female individuals across all age group has been observed in the wild to encounter with sexual exchange with other males and females across all age groups. So I applied B2 statistic on the genomic data of 13 bonobos in the grade 8 project, and here is the data map on human genome because it's easier to interpret. That is the Manhattan plot I have, and non-surprisingly, one of the highest candidate we have is on the MHC region, which is very important for adaptive immunity, and so it's great that we can find that. And across the other candidates, the other peaks that fall in the coding region, we also found a gene, KLR-D1, which encodes for CD94. It's a very important surface antigen on the natural killer cells. So we think this candidate is very interesting, not only because it's expressed on natural killer cells, but also bonobos have been observed to have very low diversity, have very reduced gene repertoire for their immunity, especially for viral defense. And some even suspect that there's some previous ancient sweeps that's happening that lead to the reduction of diversity for those genes. And here, the KLR-D1, it encodes CD94. It's a very crucial component of a receptor complex on natural killer cells that these cells are important immunity cells for especially viral defense. And we think it's very interesting. It's as if it's compensating for the loss of diversity of its binding partners or other players along the line of viral defense. And in addition to that, we also find an interesting candidates called NK3, and although we really don't know much about its function, it has a few top candidates associated with sex orientations in human males. So that's interesting. And there are a few other candidates involved in neurosensory, neurodevelopment, and innate immunity. You're very welcome to check out our preprint on bioarchive. And quickly wrap up, we developed B statistic based on the mixture model, which grant them impressive robustness to oversized windows, and they can detect balancing selection powerfully even when they're applied with least optimal window size. And we applied them on bonobos, we found well-supported, say, MHC genes, and we also found novel candidates that make correspondence to their unique biology one way or another. And lastly, we made software. It's called Bolomix. You can download it at the website here. And with that, I'd like to thank my advisor, Mike DiGiorgio, lab mates, funding source, and the amazing audience for your attention. And I'm happy for questions.
Sense for some balancing selection there too. And actually, I think it was demonstrated something similar, not exactly at the population level, but at the genome level in New York.
Yeah, we only looked, because we only examined the candidates, we didn't really went on to explore their other binding partners and relations, but that's definitely something very interesting to look at.
So you're modeling how far a window is effectively from a site under balancing selection. I'm wondering how the power changes, like if an AFS that you're building is between two sites, there are differential returns in balancing selection. Is this method going to be weaker or better at detecting those?
That is a very interesting scenario because most of the simulations and the method that's been developed here, we are assuming there's just one locus under balancing selection. And for multiple locus, then presumably there will be a much longer footprint, so increased diversity. And because this method is not really constrained by the length of the footprints, then we should have some power to pick that up, but we can always make improvements for that too.